Welcome to today's presentation. We are the data-driven subgroup of ISoP, and we wanted to have a discussion about the changes to PopPK, PD data, and some of the supplemental information that we normally submit during a regulatory submission, specifically for the FDA, and their new guidance for population PK. Now, I say new, it's relatively new. It's been out for about a year. And we wanted to at least go through some of that and have a discussion about how we might, as individual contributors to a submission, how we might go about trying to meet some of the suggestions that the FDA has made. So I will present a little bit of a disclaimer that printed at the top of each page on the FDA guidance is the following statement: contains non-binding recommendations. They go to great lengths to point out that the information that they present in their guidance is not necessarily binding, but some recommendations based on what they would prefer to see. So we will kind of carry through the same disclaimer. So the information that we're presenting here today are examples of some ways to address these recommendations, or some ways that our companies have tried to address these recommendations, and then have some considerations for what might be critical information to send in and what might be less practical for a submission. Within this presentation, if we can post it, there are links. This particular one doesn't have the full website address, but we have a slide at the very end of the presentation that contains the links that we were able to find on the FDA's website, so that you can pull up these actual guidances yourself and review them if you haven't seen them before. As an introduction, in February of 2022, the FDA formally announced guidance for population PK. It provided some additional clarification on what the FDA viewed as its best practices in both model development, model validation, and then their evaluation of PopPK. It provided some recommendations for usage of PopPK methodology, and it provided content expectations for the reporting of results. Mostly, I think it clarified some of the data requirements and the supporting documentation that they would like to see, especially around supporting evidence for any type of PopPK analysis methodology and simulations. The guidance does specifically indicate that it's non-binding and therefore very open to interpretation. I get the impression that over the years they have seen contents of submission packages that were very content intense, so a lot of information being provided to them, and in some cases very content sparse, so very little information. And so they were trying to clarify that this is what they would like to see, and please keep that in mind as we, the sponsors, create our submission packages and provide information to them. The discussion that we're going to do today covers some of the aspects of the guidance, and we'll provide some examples of how we want to meet the stated expectations, or how we propose to meet the stated expectations. As a scope of discussions, we're going to talk about detailed dataset contents, some of the supplemental files for submission that we should be considering, a reviewer supplement or reviewer's guide with some examples, some different submission summary guides, and then obviously, if we have time at the end of this, we'd like to tee up a discussion about file formats and the locations of information. If we don't have time, or we feel that this might be a useful conversation as a separate presentation, we can take that away as feedback and possibly conduct another session in the future where we specifically talk about file formats. We will, however, not be discussing clarifications of the PopPK and PK modeling strategies. When you read through this guidance, there's not a lot of new recommendations in it. It's just clarifications of what they have been given in the past and what they would like to see. I also felt that presenting a lot of information on the specifics of population PK evaluation and model assessments and model validation really should be the purview of maybe a different group within ISoP rather than us, because our focus is more the data set creation and data-driven types of evaluations, as opposed to specifically PopPK modeling or simulation. So we won't really be covering much of that at all. We'll be focused more on the mechanics of the operational aspects in the background. So with that, I would like to introduce my colleague, Neelima Thanneer. She's currently working in BMS as the head of data sciences and clinical pharmacology analysis and reporting. She's been leading the Pharmacometric Programming group over the past 15 years. She's co-lead of the ISoP PopPK Data Standards Working Group, this group, as well as been heavily involved with the CDISC efforts for ADAPPPK and the ADNCA work that's been done recently. And she also co-founded this PopPK Programming Forum. So I'd like to introduce her, and she will be covering dataset creation and reporting. Neelima, take it away, and just let me know when you would like me to change slides.
Thank you so much, Dave. I just realized we didn't introduce you, so I'm just going to go ahead and say a few things. So Dave has been, part of the PKPD Programming group, ever since I think little after we co-founded along with Jing and I. So I'm just going to give a quick bio. He's a pharmacometrician with Eli Lilly and has been with that group for 28 years. He has been involved with the ISoP and as well as CDISC, PopPK and NCA analysis data standards. And more recently, he's also part of the ADADA, which is anti-drug immunogenicity, a member of the Control Terminology Group for PK data as well. So thank you, Dave. And I'll go through some of this and pass it back to Dave for 5 formats of it. So, the way we are presenting this is on the right side, we'll give you a snippet of what is the clarification they need or what has changed from the previous guidance. And on the left side, I'm just going to give you some recommendation on approaches to address, what has been changed. So this is about reporting, right? So in the pharmacometrics report, we have several sections, and we are going to mainly cover about the data section of the report in today's presentation. So the highlighted text on the right side says, in any situation, it is important to verify that patients with missing PK data do not differ from other patients. So what this means is they want a summary of covariates of the subjects with no PK data and compare it with that they have. So in our practice generally has been, I could speak for everyone else in the industry as well. We only include subjects with PK information in the population PK dataset. So we do not include the subjects, the treated subjects that do not have PK, right? So in order to create this table, we do need to have the treated subjects without PK as well in the dataset. So that's why on the left, I mentioned that, in order to summarize covariates for all treated subjects, we need all treated subjects in the treatment groups of interest should be included in the PopPK dataset, whether they have PK or not. So to do that, how are we going to add these additional subjects because they don't have a profile? It doesn't really fit into the standard PopPK data structure where you have dosing events and PK events. So one recommendation is that we also implement this in BMS is we had to just create one record per subject for these treated subjects that do not have PK and populate the covariates, like the baseline covariates for those subjects, and flag that record or flag that subject, I should say, and exclude it from the analysis, but keep it in the data set. It can have the other variables of no interest missing, for example, timing variables, etc., as long as it has covariates populated. So that's one way to incorporate this and create a table to address the FDA's comment on, or I shouldn't say comment, the text in the guidance. So if we can go to the next slide, Dave. So here, there's a bunch of things that they spoke about, right? So one is the very beginning paragraph. It says, strong correlations between covariates can preclude the ability to make inferences about individual covariate effects and implications for the choice of covariate modeling approach. So they spoke about the covariate correlations, and they spoke about issues related to missing data including missing covariates and the data below the lower limit of quantification. They should be addressed with appropriate analysis methods. And they wanted to see something, some transparency on how we are doing that exclusion, and needed a summary of that. So on the left side, the first bullet is not really related to this, but one thing I want to mention is they suggested to rename the data section to materials and methods. However, this does not prevent the sponsor from using other section layouts and the information is provided. So if you're used to, as long as the content is organized in the correct sections, the title is not that, you don't have to, it's not mandatory to name it as materials and methods, but that was a suggestion that we noticed in the guidance. Now, speaking about the additional outputs, that I highlighted on the right side, right? One is this can be in the appendix or this can be in the body. You could do a baseline covariate table for subjects excluded. That's one inference from the guidance. And the second one is BLQ by sampling time. So there are multiple ways we could do that. We could do BLQ summary by population or by time intervals. And I'm only talking about the post-dose, post-treatment BLQ, not the day one pre-dose BLQs, because they'll be anyways, not included because they're day one pre-dose. And I'm going to show you in the next slide when we get there on how we do that. And the last one is the covariate correlation plot, the pairs plot of covariates. We have some flexibility on how we do this, but that addresses, one of their recommendation on the right as well. Dave, can you go to the next slide? Yeah, so this is the BLQ summary. We've done this in a couple of submissions, and there are multiple ways of doing this. So what we have done here, this is a summary of BLQ samples, number of samples percentage by population on the left. It's by phase 1, phase 2, 3, and non-BLQ, BLQ, and the total. And we have also done by trough versus non-trough. And we've done by sampling time as well, which is, less than, it can be, it's again, pharmacometrician will have the input on how best it can be presented for that particular project. So we are just outlining here some possible ways of interpreting, the FDA comment and then, putting this table out. Okay, and the next slide has the covariate correlation plot. Amit can, speak to it as well. He knows very well about this. So you have a beautiful plot that, visualizes the correlation between multiple covariates. This might get a bit, I think, visually complicated if you have a lot more covariates, but, it looks really good and gives the information we need for key covariates. Next one has some e-submission related topics. So that was about the data section and the changes that are needed in the data section, the last 3 slides. And this one is mainly about anything that changed in the files that we submit. So one thing, that we've stopped doing even before this guidance came out is producing the line listings. And I'm glad that they also mentioned it in this guidance. They said the appendix does not need to contain full line listings from datasets on the right side. So, if you're doing this in the past, you could stop doing this. It just doesn't serve any purpose because they have the full dataset with them anyways. And, another thing highlighted here on the right is listing of samples removed from the analysis and reasons for removal. The way I interpret is like, as long as I still don't think it's mandatory to provide the listing, as long as you have a transparent table in your body of the report that says the number of subjects or samples included and excluded, and the reasons for exclusion. For example, you have like 5, 6 reasons for exclusion, and you could present it in a table that has in each column a reason and the number of samples that were excluded because of that reason. And they are flagged in the dataset anyway. So, if you filter by the flag, the dataset will have those. But, you could provide a listing in appendix also for the samples that were excluded. And if the analysis process description is provided by the, so this is to address the comment on the right, bottom right. A diagram showing the connection between input data sets, control streams, and output data sets presented in the ER report. So I mean, there are multiple ways to do that. Some people do it in table, the flow of the analysis. Some people do it in picture. Some people do it in the reviewer's notes, that what is input to what model, and then what's the output and the sequence of events. So as long as you have a way to depict that, it should be good. It doesn't always have to be diagram, but that's something to call out here, that's a change from before. And one other thing that we've noticed recently is they are asking for scripts to create the ER data set. So for example, the parameter data set coming from PopPK exposures and merged with the response variables to create the ER data set can be submitted to the FDA along with other files. Some people I've seen submit the parameter file itself as an input, and that can be merged with the other variables. But since they already have all the files related to PopPK, they should be able to generate the parameter file. And if you give them the script to merge that with the how we have created the exposure data response data set, that should serve the purpose as well. So yeah, that's to talk about that. And the next one is more about the things that Dave is going to cover about files, e-submission. Yeah, Amit, go ahead. Question.
Yeah, so just a question and about the first slide that you presented on including the summary of subject characteristics for subjects who are not included in the
yes.
in the
yes.
analysis.
Yeah.
And the interest of really showing that these are comparable. So any thoughts of because if those subjects are simply included in the overall covariate summary, you can't tell, right? I mean, how are these different? How are they the same? So, any ideas, like perhaps maybe doing a stratified summary table with included subjects, excluded subjects.
Yes.
So you can actually do like a scan through and see a comparison.
Yeah, I think that was the intent. So maybe in the appendix, what we're thinking is to only summarize the flagged, the treated subjects without PK separately. And we already have the one with the, full covariates with no flags in the body. So we can go with that approach, or we could do side by side within the body itself of the report. But let's open it up for others' opinions as well on any thoughts on how best to address this.
Yeah, so we oftentimes have different subpopulations from various other pharmacostatistical analysis. So we have seen within a large study, for example, where there are a number of patients, only some of which may have received or be in an arm that allows for PK sampling. How we've addressed that typically with our covariate assessments is to include, sort of the demographics spread between the two groups. So we have the entire study as a whole. Our statistical colleagues often do, here's all the patients in the study, here's the patients in this particular subset group that we're looking at. And we kind of have the same type of thing where we have an additional column comparing the characteristics of patients in the PK evaluable subset of patients, as opposed to the entire population of the study as a whole. So I think you could take something like that, where you have an assessment of your covariates showing the variations amongst the population as a whole, and then subsets. And if that requires a subset of patients that are included or are not included in your PopPK assessment, I think that is something that can be, evaluated and set out and cut and displayed. It's not something that we typically have done in the past. When we have a standalone PopPK report. So you would be able to look at the clinical report and see sort of the overall population. Here's all 1,000 patients, for instance. And then in our PopPK standalone report, we might just have, you know, the 300 patients that participated in the PK sampling portion of the study. These were what were included in the PopPK data set. And I think we've left it open for reviewers to be able to open both of those files and compare them. This sort of gets at, no, we'd really like to not have to open multiple documents to look for this information. And so we'd like it presented somewhere in the report, whether it's the appendix or the main body, where you show us a high-level table that summarizes what those covariates might be, just so that we can assuage our concerns that the PK subgroup is somehow unique amongst the general population that was studied. So I think there are multiple ways that you can cut this, and obviously Neelima's suggestion of carrying through a placeholder for each of those subjects that may not have participated in the PK sampling portion, or for whatever reason didn't have PK samples taken, is a good way to handle it if you don't have easy access to some of the other subpopulation separations. So I think it's probably a good idea, if you have a standalone report, to consider showing sort of a distributional breakdown of, here's all the patients in the study, here's just the PK patients, and show that, yep, indeed, one falls within the standard deviation of the other and the range of the other. But my experience so far has been that we didn't break it down this way, that we always relied on the clinical report to show all patients, and then we only showed the subset of patients within the PK study. So this is kind of an interesting new one for us to encounter and say, oh, okay, it looks like they want that as well.
Yeah, the reason I asked the question, because it does seem like they're wanting something different. Maybe it's primarily directed at studies in which, you know, there's only a small percentage of subjects who are actually sampled for PK, and most subjects may not be. I mean, there's some people design studies like that. If you're doing exposure response, generally speaking, it's probably best to sample in all subjects, but still. There are some late phase studies where the sampling is only done for a few, and maybe it's mainly directed towards that.
I can see that being a concern. So if you did have a large phase 3 study, for instance, where you had just a small cohort of patients that had PK sampling, so you had 1,000 patients, but only 100 of them were in your PK evaluation, then that might be something that they would be concerned about. Did you somehow sample a unique group of individuals with a unique set of characteristics that were different from the population as a whole? So it's interesting, and it's something that I wasn't, until I saw this, I wasn't necessarily sure they were taking a look at, but it does make sense.
Thank you.
Mm-hmm. Any other questions before we move on to the next section? All right. Yeah, Dave,
Okay.
take it away. Yeah.
All right. So I want to focus a little bit on the impact of pharmacometric supplementary information and the types of files that we have been submitting and the FDA may be asking us to submit moving forward in the future. It's interesting because I've always wondered in the back of my mind, when we did the first submission that I remember doing with PopPK in it, which was for Lilly, was gemcitabine or Gemzar. And I remember the first time we sent them, we literally sent them everything we had. So every file that we had, we sent in. And then, of course, over time, we've pared that back. And so it makes me wonder how much of a pendulum swing the pharmacometrics industry as a whole has done with respect to how much they submit or how little they submit. And it may be a maturity thing. So a larger company that's used to doing submissions may pretty much have their processes down, and they know exactly how much they're going to submit, and maybe newer companies that might not have a lot of experience in PopPK aren't submitting very much to the agency. So what I saw in the guidance was very much a clarification of no, we really want these things to be able to do our evaluation. So keeping that in mind, I wanted to kind of go through and point out some highlights and give you some examples of how that might be handled in various ways. So one of the things that they asked for, and you'll notice that that we've made an effort throughout the presentation of specifically pointing you to which page in the guidance this came from. We thought that was a good idea rather than just saying go look and hunt and peck around in the guidance until you can find it, because sometimes it can be difficult to pick out particular sentences or sections. But one of the things that they asked for was that they wanted to make it very clear that they wanted the sponsor to provide information on the software that they use, and obviously submit electronic files that support analysis and simulation. So that's a huge piece in one sentence. It's a lot to unpack there. So this particular slide, I just wanted to focus on how we provide information on the software that we use. So within our population reports in the method section, we talk about specifically the software programs, the versions that are used in the analysis and the methodologies. So I provided some example text from one of our reports, and this also should include any special software that you've used. So if, for instance, you use R to create your data sets, or you have a proprietary software package that creates data sets, that also should be included in your method summary, for example, files. So this should not just include the PopPK analysis software that you've used, but any other supportive software as well. And along the same lines with submitting the electronic files, there are lots of potential files that we could be sending into the agencies. And I use that in plural because the differences in various agencies across the world create some challenges for us as sponsors, because we would like to think that everybody would be more than happy to accept the FDA's standard, if you will, for how to submit files to a particular regulatory agency, but they don't always, and they have their own quirks and idiosyncrasies. Focusing just on the FDA, however, they would like to see your input or control file. So whatever you call that, I gave some examples of extensions that people might have. These are not in any way to imply that you must have an input file and it has to be labeled .imp or .ctl or .mod. These are just examples. But ideally, you have a control file or a settings file or an input file that captures information around how you have established or set up the analysis software package that you're using. If, for instance, you have an automated software package that really only contains settings within the program itself and doesn't produce something like this, hopefully that program within a report might produce a section that you could extract and put into a file that would show what your input settings or parameters were. I can think of examples when we had a proprietary program in-house that did simulations specifically. So what we would provide to the FDA was our simulation settings file, which was not something that we programmed each time, but something that we had arranged to have output as a report. So that was included as part of our submission package when we decided to submit simulated information. Hopefully from your analysis, you'll have an output file. So input file, output file, your data set. And we actually submit two forms of the data set. The requirements request the data set in a SAS transport format. We also include what we call a courtesy copy as an ASCII text version or a CSV file. Mainly because we've noticed that using SAS to transport a file sometimes changes column formats, and that can be problematic. Maybe not so much with a typical software package like non-mem or even PSN or something like that. But if we are using R and we're using features within R, for instance, to read date time codes, and we have it coded a specific way, because of the translation in and out of a transport file, that can change the format that dates and times are output in, and then it will break the R code. And so when you are specifically including scripts, it is a good idea to include a copy as an ASCII text file. Some companies already submit an ASCII text file as their only version rather than a SAS transport file. Lilly currently chooses to do both just because we want to make sure that we've covered our bases. Covariate files, if your process is automated. So if you are sort of doing standalone evaluations and assessing each covariate as a separate run in non-mem, for example, you would want to submit specific covariate runs that were tied to the ones that you included in your final model. If, however, you're using a program that automates that feature, like an SCM method that you can do in PSN, you would want to submit files that are related to that process. So there are build and reduction logs that are included in PSN. So we typically submit those files as well as PSN's output report when we use SCM as a way of selecting or screening for covariates. They would like to see any script files. So as Neelima mentioned earlier, if you've used R or SAS or some other means to create a PKPD or a exposure efficacy or exposure safety related data set where you've merged in your post hoc estimates from your population PK onto a separate data set, they would like us to include those scripts as well. Or if, for instance, you've used R to do some sort of simulation or additional modeling work, they would like those scripts included. And therein, like I said before, is the issue with, does your data set come out of the untransport process in a format that your script will actually use without errors or issues? Go ahead, yes.
So there's a hand raise. No,
oh, go ahead. Yeah.
It's me again, Amit. That's
okay. Sorry, I can't see hands, so thank you.
Nobody. I could have waited much longer. No worries.
That's okay.
So I just wanted to make the comment that another solution to the potential issue of the format of an XPT file somehow being changed is to include a small, let's say you're reading in the XPT and you're really doing analysis with CSV and non-mem, because that's all it can actually accept. It can only accept an ASCII, some sort of ASCII non-mem ready input file, quote unquote. One solution to that is just to include a small little R script that says you read in you read in your XPT file and then you output that as a CSV, and if that's there, then any health authority can take the XPT file and that small little R script and generate the CSV file exactly how you had it on your system. Just wanted to make that as possible.
That's a good recommendation. Yeah.
Yeah. Sorry.
Go ahead. No, go ahead, Neelima.
No, I was going to make the same comment, Amit, at the end of it. So because I discussed this with Dave as well, like, some companies only do transport and some do only ASCII and some do both, right? I mean, at least, we have been recommending our partners that we work with also to kind of submit one data set, one file. So there's no scope of, differences. Which is mostly SAS transport file with this script to convert into CSV, if they want to, and then the name of the file of the CSV will be in the reviewer's notes on how we read into normal. So, but yeah, we could add that as a recommendation as well in the slides.
Yeah. So the last two are highlighted in red because they're not specifically required, but they might be useful if your processes that you use within your company allow you to create these types of things. So one of the things that we did early on with looking at some of the output from our population programs, the output files are often very lengthy. If you use native non-mem, the output file from non-mem actually includes the data set that non-mem read in. And in PSN, it may or may not include information that's easy to find. Oftentimes, these have key text strings that you can grep or pull in through a program to create some sort of summary file. And I know in the past, reviewers have liked the fact that we include this summary file, which is sort of a one-page high level. Here are the parameters. Here's their estimation. Here's the population. Here's the spread. Here's the range from our analysis, sort of summarizing the high level content for each run that we present. If you don't have that, I don't think it's critical, but it is something that I think a reviewer might find useful or interesting if it's something that you create already for yourself. I know most of us probably figure out ways to expedite the process and make accessing information as easy as possible, especially, like I said, when your output files are very, very long. The other thing that I over the years have got the understanding that they would like to see is any kind of tabular output from non-mem when it is used, or non-mem or PSN or whatever, Monolix, whatever program you're using, but specific output from those methodologies when it is used in a follow-along process. So if, for instance, you're using your parameter table that's output from your analysis as input into an ER type of data set, they would like to see that original table submitted as well. So some of these can be situational, some of these can be sort of courtesy type things that are submitted. I don't know that they're absolutely required 100% of the time, but it is something for consideration if you're putting your package together. So at a high level, what they would like to see, and we've received this in various ways from the FDA over the years, worded in slightly different ways each time. Usually we get very canned text in after or following a data-driven meeting or a type C meeting that we would have with them, asking specifically for these base, final, and key intermediate runs. And the way that we interpret those, obviously the base model is going to be your final base pharmacostatistic model. Your full model is going to be everything that you evaluated included. So as you build to a full model with all possible significant covariates added, and then key intermediate runs, which in theory, if you did things as separate evaluations for each covariate, they would be the runs that included the steps that were considered significant. So if in my analysis weight and weight and gender, for instance, were considered significant covariates on various parameters, I would want to include those runs where it showed that those particular parameters were significant. If you do some sort of automated covariate testing, obviously the log of that automated testing showing the significance of each of the parameters that were evaluated and picked and included and retained is probably going to be good enough. But they want some sort of way of reproducing what was considered significant and then how you got to your final reduced model. I have been asked specifically over the last few years for individual goodness of fit plots from both the base and the final models. They usually say representative set, but it's oftentimes easier for me to just dump the entire population out as a goodness of fit type set of plots that we then include as a PDF file for visual inspection. They have now requested simulation files and runs, but only if they're used for dosing claims or dosing decisions and or label claims. So if you are trying to substitute simulation for some sort of key, what normally would have been a key early phase type study. So if you're trying to say that you can simulate from your model and therefore do not need to conduct a special study in elderly, for instance, they would want to see those simulation files, whatever you're using as evidence to show that you don't need to conduct an additional study. Otherwise, if you just do simulations for some additional graphing or some additional information that you might include as a summary, it's not necessarily needed to include all of those different pieces and parts. But it seems that they have clarified, at least in this case, that if you do perform simulations and they are used for very significant things, that they do want those included in their submission as well. And then they want, since a lot of us use programs for population analysis that will only take numeric inputs, we have this disconnect between being able to carry through a unique subject identifier from sort of the CDISC world, which is very text-driven type of a variable. We can't carry that through our analysis, and so we wind up with a numeric substituted code for each individual. They would like some sort of cross-reference for that. So either a simple table that says, here's my subject ID, here's that I use in my analysis data set, and here's the eSub ID that you're used to, that links it to all the other information out there in the submission volume. Or you remerge into your output at a later date, or include a separate file with and without that, just so that they can make that linkage, just so they can map it back to the eSub ID for other information within the study. And I think it's the last piece that I've interpreted in this is my little box in green there, which basically says they would really like the ability to link data sets and submitted model files to the analysis and to the conclusions. So the more information you can provide them, the better. Whether that information needs to be provided in the report itself or in a supplementary guide, that's kind of up to the sponsor to determine. From a best practices standpoint, though, it should be included somewhere. And then obviously,
I have a question.
Go ahead. Yes.
Regarding the previous slide where about the model files needed for simulations, would that also include VPC?
We typically include VPCs in our report, but I have not to date included the actual scripts that were used to create those VPC plots. So if, for instance, I read my data set and simulated output or my post hoc estimates into an R script, and that did my VPC plotting, I have not typically given them the script that did that VPC. I think our assumption is that that sort of model evaluation is standard enough that they should be able to do it if they wanted to. I have never had a reviewer come back to me and ask me for those specifically. So as a roundabout answer to your question, I've never been asked for it personally. That doesn't mean that they wouldn't desire to see that. And it doesn't mean that you couldn't include those files. They probably would accept them. I don't know if they would actually ever use them. I think some of this is we want all information at hand in case we don't agree with your conclusions, but if we don't see a reason to disagree with your conclusions, we probably aren't going to dig too deep. But as of right now, in the whole time that I've been doing this, which has been quite a while, we have not had a reviewer come back and ask us for the actual script that created the VPC plots. Does that help?
Yes, thank you.
Okay. Mm-hmm. So results from analysis, methods and codes, they've asked for separate files from the study report. So a long time ago, we used to have a practice of putting all of our output into the population report in an appendix rather than give it to them as electronic files. Here they are putting their foot down and saying, now we really don't want to have to dig that out of the appendix. We would really like it as a separate file that we can evaluate rather than having to try to copy paste stuff out of the appendix to run as a script. So if it's a model file or if it's a script, you're going to be submitting that as a separate file. As Neelima pointed out before, they would like to see a listing of samples or patients removed from analysis and why. A lot of times we've interpreted that as a table that we would put in the appendix. I have strictly interpreted it recently as I want to see the patients that you have censored from your data set that actually had measured concentrations or measurable concentrations. So if, for instance, a patient came through and had all BQLs, I don't think that's as critical as a patient that came through and had concentrations that you decided to omit for one reason or another. Say it was a pre-dose sample for an exogenous compound that showed that the patient already had drug on board. So likely there were issues with the dates and times that were written down in the source data set so that you get this anomalous value being presented. If you're taking that out and it's a measurable concentration, they want to know why you've done that. And again, I don't know that they would ever have cause to question one or two of those values being censored out of the data set. They just want to make sure that there's not a practice that is egregious enough that it might actually be skewing the way your analysis flows or the conclusions from your analysis. Run records describing steps in model development. So that's something that we haven't produced in the past, and something that we're considering now is placing it in the methodology section, sort of including or do we include it in the reviewer's notes? So here's what we've given you. Here are the run numbers or the run sequences that were used to produce that. That particular piece, I'm not sure how useful that's going to be for a reviewer, especially since there are really no permanent conventions that any one company might follow to say that, yes, the very first run that I make is going to be run number 1, and the very last run that I make is going to be run number 100. And you can assume that anything in between is a step along the way. Obviously, that would be a best practice, that things would be sequential, but obviously things might not necessarily be sequential as we kind of go back and forth through and do our evaluations. So is it really critical that we present some sort of overall visual workflow that says step A is this, step B is this, and these are the numbers associated with it? I don't know. I don't have a good feeling for that. In our reports currently, we have sort of a visual representation of our overall methodology of how we develop models and then how we do the build and reduction steps and what criteria we use to select from that. But that's sort of a generic workflow, and it isn't modified specifically to address the analysis that we do for any one particular evaluation. One of my colleagues had interpreted this as, okay, in my reviewer notes file, I'm going to add in additional information that we're providing with the files that we are sending. So the base model files, we're going to give them the run number from that, and we're going to give them the objective function that came out of that and how it changed. Obviously, those things are interpretations, and they may or may not be useful for the reviewer. I don't really have a good feeling on that one. So this particular step seems a little problematic, just based on for us to be able to produce and provide consistently to the FDA, just because I don't think there's a lot of good background on what they're looking for and why they want it. If you already do it, great. If you don't do it, I guess we'll see if they start really pushing back in the future and saying, no, we have to have this, and you didn't include it, therefore I'm not going to review your submission. So this might be one of those nice to have rather than need to have sets. I guess we can all interpret that as we want to. And then the last thing was this diagram showing the connection between input data sets and codes and control streams and output data sets. And they suggested that it be presented in the exposure response report or the standalone PopPK report. And my immediate response to this was, well, will a tabular format suffice? Because I'm not sure how easy it is to create this sort of visual diagram that shows things. But if I've got a table that shows you, here's my base model and here's the files that are included, is that good enough? And right now, that's how I'm interpreting that. So again, we'll see moving forward how insistent they are on having these things covered. As before, there's the disclaimer at the top of their guidance saying this is just a guidance. This isn't a prescriptive answer to how you should put your submissions together. So your mileage may vary with respect to which ones of these you consider to be critical to a submission and which ones you don't. So this concept we dropped a couple of times of a reviewer notes file, and we didn't always include reviewer notes, but a lot of times we thought it was helpful to include, specifically to walk the agency through what files we're submitting, because sometimes they can be very numerous. And knowing that each company has their own nomenclature for how they label and name files and use files in a submission package, I think it's useful as a practice for probably everybody to have some sort of supplemental file that walks the reviewer through the additional information that's being presented outside of the population PK report. So what do you discuss and where do you discuss it? And so they have noted and allowed for the fact that this can be accomplished by providing a definition file or for information or a reviewer guide or the codes actually utilized in your analysis and data set assembly. So what I'm getting from that is we want some sort of way of mapping this. However you provide it to us is fine, as long as the reviewer can go through and figure it out and figure out how you've done it. And so leading us by the nose is probably not a bad thing.
I had one more thing, Dave, here. So yeah. One thing that helped in my case is we've been submitting the analysis dataset specification as part of an appendix. Also, like a full detailed specification that includes everything, including the rules for imputation and whatever assumptions we made. notes, and that also helps for transparency.
Yeah, agreed. So I kind of think that the methods, the logic, the workflow should be discussed in the report, either in a diagram format, as I mentioned before, or as a narrative. I don't know that it necessarily needs to be re-discussed in a notes to reviewer file, but that is a possibility of what could be in a notes to reviewer standalone file. Obviously, information linking row numbers to analysis groups or run numbers to analysis groups. So what is the final model? What is the base model that I'm submitting? What are the files that I'm submitting that cover your requirement for significant covariance? A discussion and listing of excluded data. Obviously, we typically put that as a table in the report, but it could actually be as a separate file. Or, as Neelima mentioned, if you have those things as flags in your analysis data set, which will be part of the new PopPK standard, ADAM standard for population PK, are they even needed in a tabular format anymore? If I've got a clear definitions file that tells you what those codes mean and they're flagged there, is that enough? Or does the FDA really want to still see it as a visual representation in the report? I think those will be questions that maybe will be answered in the future once people start submitting in that format and try to push back and see how much of this is again, nice to have versus need to have. And then what could you put in a reviewer notes file? So I think personally, what should be as a minimum in that reviewer notes file is the definitions of file types, and specifically what my file naming nomenclature is for my particular company. What do we call things? What do I call my control stream or input file? What do I call my output file? What do I call a settings file or a summary file? Or anything additional that I choose to include. Anytime I change names of a file from its original source. So despite as many admonitions as I have made to my fellow pharmacometric colleagues of being conscious of how they name files and how verbose they are in the naming of their files, we always seem to get to the end to a submission time and have to change names, either to force things to lowercase, include underscores where they've included dashes, or underscores where they've included spaces. Usually spaces aren't a problem so much anymore. And then certainly things where we've changed, again, a CSV file to an XPT file, et cetera. So I think there should be some sort of tabular representation of here's the original name, here's how it got changed in the submission. And oh, by the way, this is what this file is. Organization of the file lists by analysis stage or grouping, defining what each file is, the run number, et cetera. And then a high-level description potentially of analysis. So you could have an introduction section in your notes to reviewer file that basically is a high-level summary of what you already presented in your PopPK report, just to kind of remind the reviewer, hey, when you're looking through this, these are the analysis that we did, and here's what we're including within our submission package. Could it include more? Absolutely. It could include a lot more information. And like I said, I've had colleagues that have created them and decided to include things like their objective function changes between each of the sets of files that they are presenting. I've had colleagues who would like to include things like their the specific run numbers or what sequence that was in their evaluation in the file. And I think there's accommodation for all of that, and you can use all of that if you so choose. So an example that I wanted to provide is what I meant by this file extension format. So we always provide this with our notes to reviewer files as sort of the upfront, here's our naming convention. So anytime you see a file that's submitted with a .imp, for instance, that will be a control stream or an input file. If you see a .op, it will always be an output file. If you see a CSV, it's always going to be our data set. We've got this concept of a summary file, and it's always going to be .sum. And then PDFs, if we give them to you, are generally always going to be graphical reviews of the data, whether they include the diagnostics plots, our VPC plots, or as I mentioned before, recently I've had a lot of requests for individual fits. So showing the population estimation and the actual data overlaid on top of each other for both the final base model, so the final structural model, and then the final covariate model that you decide on. Just, I think they want to see the difference in how the fits have been adjusted. Did you actually improve that through your modeling process? So this is kind of a table that we present right up front and first within our reviewer guide that gives a high-level definition. When you see this extension, this is what type of file you're getting and why we're including it. Similarly, I like to include a table that shows data sets. So here's the original data set name. Here's how it got changed because of either naming conventions in submission packages or the fact that it was changed to a transport file. Or, for instance, I was really verbose and I exceeded some sort of file length limitation and we had to truncate that file name. Certainly when we go to extremes, a long time ago, we used to be told, hey, you can only have 8.3 character format for your file names. And so obviously we would have these really long file names because we typically did our analysis on the Unix side. Unix didn't care about the file length name. But when we went to submission, we were told we had to constrain everything to 8.3. And so then a table like this was very important because the very cryptic 8-character limitation that I was given had to be mapped somehow back to the actual file length name. And you're right, the suggestion that was made of having a script that might consume those transport files and then spit out a properly named and formatted analysis file might have been a good solution at that time, but that's not something that we did when we had to reduce those file names by a large amount. I wish that I could get all of my colleagues to just kind of be good corporate citizens and keep their names in the right convention, but somehow that always isn't the thing that's at the foremost of their mind when they're going through the modeling process.
So one more thing to add here is, doesn't matter what the CSV file name is, right? When we prep it for eSub, so we will use SAS transport 5 engine, and then it only requires 8 characters. So you are naming it within 8 characters, which is also the new CDISC convention, right? So we are all going to, change for those who have not already been doing that to change XPT names within that. So I'm not sure. I think the transport files that are being generated outside of SAS, maybe with R, only allow these long names, but SAS does restrict to 8 characters. And as long as, we provide a script or put notes in the reviewer's guide, this was changed to that data set, it should be good.
Right.
One more thing, can I add, Dave, in the previous slide, yes. Slide 17? Yeah. Mm-hmm. So here, FDA has a few years back has started accepting files in their original convention, like, if it's .ctl or .lst, you can submit the way they are. You do not have to convert it into, -r.txt, for example, if it's an R file. But, internal company systems had trouble, changing their software or tools or backend to accept these multiple formats, and that's why companies are stuck to submitting, still converting to .txt. But it's possible, at least with CEDAR, they do accept, and if you can work with your company, the groups that are managing these systems, you could submit the original file, and we were able to successfully do that in BMS and submit original extensions.
Yeah, that is absolutely true. And I one of the things that, at least for right now, has been a sticking point within my company is that CEDAR seems to be the only portion of the FDA that has agreed to allow sort of this native format and native naming. CBER has not. So CBER still seems to be sticking to the original guidance that was issued with, here's the files that are allowable for electronic submission formats. And that literally is transport files, PDF files, and .txt files. Mm-hmm. And so because the two sides of the FDA haven't come to an agreement or haven't issued a joint statement saying everything is equivalent or fine for submission to both houses, they don't want to have to make that distinction, and so they're not willing to work with me currently to allow those sorts of files to be passed on in their actual format. Interestingly enough, the PMDA takes a completely different view on this, and they don't want things changed at all. And so they don't like transport format. They don't want things in SAS transport format. They want the original files as they're created as CSV files. So we're already doing sort of those sorts of things for the PMDA, but my regulatory group does not want to budge until they actually see it as a published guidance or mandate from the FDA that this is okay to do this. So if you can get away with it, great. I would love to be able to as well, but at least within our company, we're one of those cases where our regulatory submission group is still kind of stuck with what they feel is the way that it should be done. And that's why I made a note here in red at the bottom that currently it's CBER that's suggesting or accepting files that have not been renamed and actually have text out on their website saying it's okay to do this, but CBER has not.
Yeah. Amit, did you have something to say? I saw your hand go up and down.
Oh, no, yeah, no, I was just going to ask a minor question about uppercase and lowercase, but it sounds like it's a bigger issue between CBER and CDER. I was just going to ask whether, uppercase R markdown is allowed.
I think so.
Yeah.
Go ahead, Dave. Yeah.
I was going to say, as of right now, I'm still being forced to submit everything in lowercase. Our regulatory submission group has created programs and created processes where they read in file names or prep those file names for the submission volume package, and their systems force everything to lowercase. So unless I acknowledge that and sort of change things on my end, I have no idea what they're going to be called when they wind up in the submission volume, unless I go back in and audit that before it's actually submitted to the FDA. So again, while it's possible that the FDA will accept uppercase in certain cases or in all cases now, I'm unfortunately still stuck in the dark ages when it comes to
it's like uphill battle with the internal regulatory group, right? I mean, it took me years to get them to, accept the original format, but you're right. But only a subset of submissions are going to CBER, right? Most of our pharmacometric reports are being submitted to CDER only. And I did bring up this with Jeff Florian, our friend at FDA. And then they're going to discuss and come up with a joint, hopefully, statement that the CBER and CDER will have same requirements, but that hasn't happened yet.
Yeah, and I think the other point that I had heard when we were having that conversation with Jeff was that even within the FDA, there seems to be a disconnect between groups. So the file submission group, the group that sort of handles and manages our eData package that we send in, doesn't necessarily talk to the pharmacometricians. So when we had this conversation when we were wrapping up in our work on the ADAM PopPK standard, that particular group came back with a bunch of questions, which all surrounded or all focused on what is this and why are you submitting it? So it's clear that they don't necessarily communicate within the FDA, just like we have disconnects between departments and groups within our own companies. So hopefully we'll get to a point where they can work through this, and then maybe we don't have to make many changes or any changes at all to the naming of our data sets and the extensions. That would be nice. That would be a nice place to get to. Because as of right now, as I mentioned before, the PMDA has a different set of criteria. Brazil has a different set of criteria. Australia has a different set of criteria. Canada has a different set of criteria. They all just won't submit, for instance, like the FDA or accept the FDA package. And so we have to make slight modifications in everything that we submit to these different regulatory agencies. It would be nice, A, to get the FDA to get their shop in order, and then to be able to go around to the other regulatory agencies as well and say, hey, this would make our life so much easier if we could all agree that this was sort of a standardized way of submitting data. We've sort of done so with, our standards for reporting. Why can't we do so with our standards for data submission? And then I think this might be one of the last ones I have for examples for the reviewer notes file. This is would be for me a specific section. So I'm talking about just the files submitted for the base model. And so again, I would go through and I would say specifically, here's my input, here's my output, here's their names. And as Neelima pointed out, in my case, I have changed them. So I've replaced the .op with an underscore and then added a .txt to the extension. And of course, in my reviewer notes file, I have to point out that in order to make these able to run, they have to remove the .txt extension and rename the last underscore to a dot to be able to use these as files. And that is why I specifically provide the original file name and the name used in the submission so that they at least have a reference of what to go back to. And then simulations, I wanted just to briefly address that. So the comment in the guidance was, when appropriately justified, submission or simulations is what they're referring to here. Such analysis in conjunction with ER data have been used to approve dosing regimens that have not been directly evaluated in clinical trials. So I think they are keen to see simulation data when it is being used as a substitution for a clinical trial, or when it is being used as a justification, as a standalone justification for dosing or formulation decisions or those sorts of things. So when it is critical to your submission package, then absolutely they want to see simulations and presumably the scripts used to create those simulation files for the input and output. So that's when we should submit them. What data should we submit when we submit simulations? Obviously, the input data file. So the covariate files are information that we use to set up the simulation, the code itself, whether we're doing it in R or some other type of program, and then obviously the simulated output. So they want to see those sorts of things included. And I think ultimately the goal of the agency is, can they rerun that simulation, or at the very least understand how to reproduce it? But again, wanting to make the process of reviewing our PopPK data and our exposure response data and any sort of simulation data as easy as possible. I think providing them with everything they need to push a button and rerun it is probably a best approach, rather than assuming that they can do their own work and somehow reproduce what you've done. So we have a little bit of time. If it's okay, I want to cover the additional information. Again, this might be a topic for its own discussion, but we at least wanted to get it out there because I felt like that the comments came up in our presentation, at least our notations of differences in file naming conventions that are accepted and what agencies can accept which. And so I did put a file or a slide together that talks about the file formats and what is accepted, or what at least has been noted by CDER versus CBER. So the two main pages where you find this information, the first one is the link to the model data format page. And then the second is the link to the official specification for transmitting electronic submissions. And those two pieces of information get you to a set of files that sometimes is congruent with each other and sometimes incongruous. So sometimes there are discrepancies in one file versus another, which is why I feel you need to include both. The link at the bottom was part of a footnote that I decided to include. It's almost a circular link. So you click on that, you get this other, you click on that, you go back to the first link. And so I'm not sure that it's that helpful. But the first two links are where I got the information that's presented in the bulleted list in this slide. And so specifically, it talks about whether or not you need to include SAS transport files, or if comma delimited is okay. And that's listed in the page 4 specifications, and CDER has indicated in that document that they're willing to accept CSV as a way of submitting a data set. It almost sounds to me, again, I think we need clarification, but it almost sounds to me that they would accept that in lieu of sending it in as a SAS transport file. But again, I don't know if their data group agrees with that. So that's something that I think we need to clarify. Description of each data set and associated variables can be provided in a definitions file. So that's in the model data format section. Reviewer's guide is talked about in the model data format section. Model codes and control streams and output listings for all of the basic structural steps, that's covered on the model data format page. Files for generating key tables and figures in the report, and relevant simulations can also be included. And they talk about that they can be submitted as ASCII text files, But they also specifically point out that it's not necessary to rename those ASCII files ending in different extensions. So this is the first place that I saw it actually called out that, hey, you don't need to do this .txt extension. That's on the model data format page. However, in the specifications document, it specifically says you can only submit them as this, and only CDER takes it this way. So again, I think this is a CDER specification only. And then CDER has also gone on record in the specification and say that they are willing to take our markdown files and Phoenix project files, which are actually encrypted zip files. So in the past, we have not been allowed to submit zip format or a compressed format file, but it looks like they're willing to at least accept that. And so maybe some companies have requested or tried to submit their NCA analysis or maybe even individual modeling of data in Phoenix as a project file. And then I know that there have been recent exceptions for SimSip for things that have done with PPPK modeling. And so there's a specific guidance for PPPK modeling where they call out the fact that they don't want you to rename the files. They want to be submitted as the proprietary files that go, for instance, with SimSip. So I think there's a lot of confusion out there. I've tried to identify where the best places to look for those, but obviously the FDA websites are an ever evolving thing and ever changing. And so the links that I provided here and on the next page are as of June 2023. So that's my disclaimer. I could find them at that date. Hopefully they all still work if you go to use them. But obviously as things change, these links may or may not be the most updated versions of things. But I provided a link for that file format specification as a PDF file, the FDA standards catalog, which is currently an Excel file, the model data format page, which is basically just a narrative discussion page, and then obviously a link to this guidance that we're talking about specifically today. And then just as a comparator, I provided a link to the current PMDA guidance for pharmacodynamic and pharmacokinetic analysis. It's a translated version that's out there on their website. So with that, that's all I have for this presentation.